So I wanted to go over today in this short revision video the cell cycle and mitosis. So when we think about the cell cycle itself, we can think of the cell cycle in terms of a circle, which can be split into two parts. You've got the small part, which is here, and then you've got the larger part of the cell cycle, which takes the majority of the time. The larger part of the cell cycle, taking the majority of the time, approximately 23 out of every 24 hours, is interphase. And this small part of the circle, that is the actual process of mitosis and cytokinesis, the actual division of the chromosomes and the splitting of the chromosomes, the cells, into two. Within interphase itself, you've got three main phases: G1 standing for growth 1, S standing for synthesis, and G2 standing for growth 2. Now there's not much difference between G1 and G2. Both of them are preparations for cell division, although G1 is more about growth and G2 is more about the actual preparation for the actual mitosis. But the principles are the same in that you will get ATP synthesis happening. You'll get lots of protein synthesis happening. And you'll get organelle duplication. And that's going to be much the same in G2, the only difference being that you definitely get the duplication of the centrioles in G2. S phase is specifically DNA replication by the semi-conservative method to double our DNA molecules within the cell. So if we then have a look at an actual graph, and on this graph we will plot two axes. Obviously, our y-axis will be DNA content in a cell, or it could be in a nucleus. Sometimes it's one, sometimes it's the other, in what we call arbitrary units. And we have time in hours along the side. We've got a little bit of a scaling, 1, 2, 3, 4 for our arbitrary units, and we'll do our time in 5-hour intervals, just to give us an idea of what's happening. So if we look at the graph, it's starting off at 2, say, and it will continue on that way quite happily. Then it will double to 4, and it will continue at 4 for a bit longer. Then it will reduce very quickly back to 2. Then it will stay constant again before it starts increasing again and the cycle continues. So if we look at the stages, over here we have G1, we then have S phase, this is G2, this is mitosis and cytokinesis, and then we start again with G1 and S again. So we've got the doubling of our DNA to double the content. So if we'd had 24 molecules of DNA, we would now have 48. And we'll come back to that in a minute. Now, sometimes you're asked to work out the length of one cycle, and it's really tempting to just go, oh, it's from there to there, which would be about 16 hours. There's a problem with that. Often, they won't start the graph at the beginning of, say, G1. So it's much better for us to look from repeating unit to repeating unit, for example, from there to there, and work out how much that is, because that is also one cycle. As you can see, from there to there, that's, say, 26 hours minus 7 hours, you've got 19 hours, okay, which is actually longer than we would have got if we'd done from the beginning to the end. So be careful with that, please. That's the same with any repeating unit. Okay, let's look now specifically at mitosis. Now, we know that we start with a diploid cell and we end up with two genetically identical diploid cells. Now, the numbers don't work there, as you can see. So what must be happening in the middle is we must be doubling our chromosomes so that they can then split into two. And that's what's happening during S phase of mitosis. Now, because all of the cells produced are genetically identical and all diploid, we use this in growth, repair, and asexual reproduction. So let's look at what's happening in terms of the actual chromosomes, because that's the trickiest bit. So if we start with a cell, the beginning of interphase, and let's look at the chromosomes that are in it. So I'm going to put a nuclear membrane in because it is an interphase. I'll make it fairly large so I can draw inside it. Okay, and I'm going to draw in 4 chromosomes only. So I'm going to do 2 nice big red ones and 2 smaller blue ones. So this is a 2N, a diploid organism where 2N equals 4. And we have 4 molecules of DNA here. And we have 4 chromosomes. Now, we're now going in through S phase, and during S phase, each DNA molecule, as we know, if this is one DNA molecule, one chromosome there, each DNA molecule will duplicate itself in semi-conservative replication. And you'll have 2 identical copies, but they're held together by a centromere. So we have, if we look in terms of the picture, we have our cell again, like so, with our nucleus inside it. But now if we look at the chromosomes, what we actually see is they're looking like this. 2 red ones, and 2 blue ones. We still say that they have 4 chromosomes in them, but now they actually have 8 molecules of DNA. And the job of mitosis will be basically to separate these molecules of DNA so that one goes to each pole. So we call this whole thing is still called a chromosome, and that's really important. The thing that holds them together is called the centromere, and these are called sister chromatids. Now, let's look at what's actually happening then. So, always useful to remember, IPMAT. I've remembered that since I was learned it myself. Interphase, prophase, metaphase, anaphase, telophase. Now, we've talked all about interphase already. So let's just think about prophase, because prophase is where the interesting things start to happen. So here is our cell. Okay, first of all, you'll notice I'm not going to draw a nuclear membrane. So the first thing we can say is that the nuclear membrane disappears. Okay, now the chromosomes during interphase were actually very long and uncondensed. And what happens in interphase, in prophase rather, is that they condense down. And when they condense down, they get seen as these sister chromatids connected in their centromere, randomly placed within the cell. Okay, so we describe it as the chromosomes condense, and that's what you're used to seeing when you draw pictures of chromosomes, to form two sister chromatids connected by a centromere. Now, the other thing that will happen is that our centrioles, which are responsible for producing our spindle, they will migrate and they will go to opposite poles. We always draw them as two tubes at right angles to each other, and they will start to form the spindle fibers. Okay, so centrioles migrate to the poles and form spindle fibers. Now, that's pretty much the end of prophase. So prophase was a busy time, much busier than some of the other stages we're about to look at. So if we move on from prophase, our next stage is going to be metaphase. So once again, we have our cell. It's got no nucleus. What it does have now, we'll stick to the same color scheme. What it does now have is our spindle that have formed across the cell, like so. And the chromosomes will line up along the center, the equator, in any order they like. I just happen to draw them this way. And their centromeres will attach to the spindles. Okay, so chromosomes line up along the equator of the spindle, attached by their centromeres. Okay, so that's pretty much the end of metaphase. It's not a very busy phase, and you can really see that when you look at an electron micrograph drawing of mitosis, or when you look at a light microscope drawing of mitosis, you'll really see the equator. So in prophase, you'll start to see the chromosomes. Here, you'll start to see the equator. Bear in mind that sometimes, however, they give you a picture where you're looking from the top. If I were to look at the top at these chromosomes, it's in 3 dimensions. So what I would actually see from the top view is I would see that, them all sort of circled around. Okay, that would give me my top view. Just need to bear that in mind. Okay, so after metaphase comes anaphase. So here is my cell again. Here are my spindle. You'll notice I don't draw them completely to the end this time. Okay, the centrioles will still be there. Okay, and what you've noticed happened is that the spindle has actually broken. And when the spindle's broken, it's because the spindle fibers have contracted. And when they contract, they break apart the sister chromatids, breaking the centromere and pulling the sister chromatids to opposite poles. So we describe it as the spindle fibers contract, the centromere breaks, and the sister chromatids are pulled to opposite poles. And that is basically what you'll see in anaphase, and you'll see it quite clearly in a drawing or in a light microscope. That leaves us with telophase, which is the last phase. Okay, in telophase, we've got our cell, but this time we have our nuclear membranes reforming, 2 of them. And your chromosomes are basically start to decondense again. They're back in their individual, there and there. So the chromosomes unravel again. They decondense. But you'll notice there's actually no split. That split will have to come in cytokinesis. So although this is telophase, telophase just means we've now got 2 nuclei in one cell. We're now going to have to actually split those nuclei into 2. And there's 2 possibilities, depending if you're an animal cell or a plant cell, okay, for cytokinesis. If you are an animal cell, then you form what we call a cleavage furrow. Okay, it literally squeezes in like that. Imagine getting a piece of plasticine and sort of squeezing it until you manage to separate it out into 2 balls. That's basically what happens. So we say a cleavage furrow forms. And this is specifically for animal cells. This won't work with plant cells, because if you think about a plant cell, okay, which obviously I've drawn animal cells all the way along, but if I think about a plant cell, the plant cell's got a little bit more of a problem, because the plant cell has a cell wall going around the outside. It's not really possible for it to do a cleavage furrow, because if it does a cleavage furrow, then it would need the cell wall to be flexible, and the whole point is that the cell walls are made of cellulose, and cellulose has high tensile strength, and it's not, although it's a little bit flexible, it's not flexible enough to bend all the way. So there must be another method, and their other method is actually that they build their cell wall from the middle outwards like that. Okay, I'm going to put an arrow. Okay, and then we say the cell wall builds. You'll see this called a cell plate. And various other things, but that's the basic principle. So that by the end of it, we have 2 cells, each genetically identical to each other, and each with a diploid number of chromosomes. I hope that helps. I'm going to make a different video which will cover meiosis specifically. If you've got any questions, please feel free to ask.